Hello dear children, welcome back. I'm Dr. Jisha Joe. In this video, we will be going through the important focus points from the chapter Structure of Chromosomes, Cell Cycle and Cell Division. These are the most important topics from which you can expect multiple choice questions for your first semester board exams. At the beginning of this chapter, we have the two terms, the chromosomes and the chromatin. You should know the difference between chromatin and chromosome. Chromatin is present in the interphase nucleus, and we know that it is these chromatin which condense and coil to form the chromosomes as the cell enters into the cell division phase. Then coming to the discovery of chromosomes, it was Walter Fleming who first studied the chromosomes in animals. Then next topic is chromatin. In this topic, you should know what are the basic constituents present in the chromatin material, what is the composition of these constituents, that is the DNA and histone proteins, 40% DNA and 60% histone proteins present in the chromatin. That also should be known to you. Then moving on to the molecular structure of DNA, you should know who was the person who first studied the shape of DNA molecule. It was Rosalind Franklin. Then what is the contribution of Watson and Crick? It was these two persons who proposed the double helical model of DNA. That also should be known. Then the basic structure of DNA, the DNA molecule comprising of the two strands, what are the repeating units of each strand called, that is the nucleotide. What are the components of the nucleotide, that is the pentose sugar, the nitrogen base and the phosphate group. What is the pentose sugar that is present in DNA? It is the deoxyribose sugar. And what are the bases present, nitrogen bases present in DNA? Adenine, thymine, cytosine and guanine. Now, what is the complementary bases that are present in the DNA molecule? You should know that adenine always base pairs with thymine. And this base pairing is by two hydrogen bonds, and cytosine forms a triple bond between cytosine forms a triple bond with guanine. So adenine pairs with thymine, cytosine pairs with guanine. That should be known to you. Then you should know the basic structure of a nucleotide. What is a basic structure? Nucleotide is composed of a nitrogen base and phosphate group and a sugar. And the two strands of the DNA can be given, and it can be asked as a diagram based question. That is, the two strands of DNA can be given and you will be asked to label the different parts of the DNA strand or the DNA molecule. And questions based on that can be asked, like for example, what is the number of hydrogen bonds present between adenine and thymine, which are the nitrogen bases, what is the sugar present in DNA. So any different type of question related to that can be asked, any related to the structure of DNA can be asked. Okay, and don't expect any questions from outside our syllabus, just give importance to what is given in the text. Okay, be thorough with what is there in the text. Okay, nothing from outside can be asked for at the 10th standard level. Then moving on to histone proteins, you should know that histones are proteins. They are proteins, and what is the function of histones? They help in coiling and packaging of the DNA into units called nucleosomes. So nucleosomes are the basic units of the chromatin material. And what is the structure of nucleosome? How many histone proteins form the nucleosome core? You should know it is the histone octamer. 8 histone proteins form the nucleosome core. Central core is formed by 8 histone proteins around which the DNA strand is wound. So all those things should be familiar to you. Then moving on to the structure of chromosomes, you should know a duplicated chromosome has 2 arms which are called as the chromatids attached to the centromere, and you should know the function of the centromere. Okay, that is important, function of the centromere. You should know the difference between centromere, chromatid, okay, chromosome, chromatin, nucleosome, nucleotide. All these might be confusing to you, but you should know the basic difference between all these terms. Then moving on to the next topic, what are genes? You should know the definition for genes: sequence of nucleotides on the chromosome that can code for a particular protein, that is genes. That is a gene. Now, moving on to the next topic, what is the need for the formation of new cells? There are four purposes are there: for growth, for repair, replacement, and reproduction. The first three, that is growth, repair, and replacement, involves a type of cell division in which the chromosome number remains the same, which is called as the mitosis. And the fourth type, that is the reproduction, in lower forms, in unicellular forms, the type of cell division taking place, that is in bacteria and all, you can see the cell chromosome number remains the same. So it is just the mitosis that is taking place in unicellular forms, whereas in the case of higher forms, higher organisms like human beings, large trees and all, or higher plants, higher organisms, you can see that the cell division is taking place during the formation of gametes is meiosis, where the chromosome number is getting reduced to half. So that is important there. Now there is a box showing the number of chromosomes in humans. Each and every cell of the human body has 46 chromosomes. During gamete formation, we know gametes are produced inside the testis. The testis produces the sperms in males and the ovary produces the ovum or the egg in the females. So during gamete formation, what happens, this 46 is reduced to half, that is 23. So the sperms contain 23 chromosomes. Each sperm contain 23 chromosome. Each egg produced also contain only 23 chromosomes. The fusion of the sperm and the egg produces the zygote, which has the 46 chromosome number, or 23 pairs of chromosomes. So this is meiosis taking place. So the type of cell division that is needed or that occurs during the formation of gamete is meiosis, that you should know. Now, what is cell cycle? You should know what are the phases of cell cycle. The interphase and the M phase are the two phases of cell cycle. Interphase is the non-dividing phase, and the dividing phase is the M phase. You should know that. Now, what are the different phases of interphase? The G1, S, and the G2 phases, and what are the things or what are happening during each of these phases, you should know. Okay, sometimes you might be given options to choose which is not happening in a particular phase, or which is that phase, which is that stage during which the synthesis of DNA occurs. So you should know that it is during the S phase that DNA synthesis or the chromosome duplication take place. So you should know the basic difference between all the three phases, the G1, S and G2 phases. So that is important there. Then moving on to the next topic. So there are a few cells, you can see certain points given there, that is the brain cells. You can expect a question like that: cells which do not divide further once they are formed in the embryo, they do not further divide. Which are those type of cells? They are the brain cells or the nerve cells. Okay, so once these cells are dead, they cannot be replaced. But in the case of liver cells, the skin cells, meristems, etc., they undergo division. Germinal cells, etc., they undergo division. Then uncontrolled cell division occurs. That also should be known to you. Uncontrolled cell division occurs during the formation of tumors. So that is also important. Okay. Then coming to the cell division part, we know two types of cell division which I have already discussed before: mitosis and meiosis. Mitosis, chromosome number remains the same. And what are the phases of mitosis? You should know what are the phases. There are two phases in mitosis, that is cytokinesis or two stages: cytokinesis and karyokinesis. Karyokinesis and cytokinesis. Karyokinesis involves the division of the nucleus, and cytokinesis involves the division of cytoplasm. Now, what are the phases of nuclear division or karyokinesis? So, mitosis involves both karyokinesis and cytokinesis. Okay, and all the nuclear changes come under karyokinesis. It is the division of the nucleus, the nuclear material. So what are the phases coming in karyokinesis? Prophase, metaphase, anaphase, and telophase. Okay, so M phase or the phases of mitosis include prophase, metaphase, anaphase, and telophase. So you should know each phase of the mitosis. You can expect a diagram based question from that part. Okay, any phase can be given. You might be asked to identify the phase. You will be asked to identify the number of diploid chromosomes in that cell in the figure. Which is the stage that is prior to this phase, or which is the stage coming after the stage that is given in the diagram. So such questions you can expect. You can expect one diagram based question from any phase of mitosis. It may be prophase, metaphase, anaphase, or telophase. Okay, but since we have just MCQs, so you need not draw and study, but you should know what are the changes happening in each phase of mitosis. So we know during prophase it is the centrioles, the duplicated centrioles start moving to the opposite poles. The chromosomes become distinct. The chromosomes have already duplicated during the S phase, and the chromosomes, each chromosome will have the two chromatids attached at the centromere. Spindle fibers begin to appear from the two centrioles. The daughter centrioles which have reached the opposite poles start to produce the spindle fibers, and the nuclear membrane and nucleolus start to disappear. By the end of prophase, the nuclear membrane, nucleolus have all disappeared. So these are the things that are taking place in prophase. Metaphase, what happens? The spindle fibers which are attached to the centromere of the chromosomes bring the chromosomes and align the chromosomes at the equatorial plane of the cell. Okay, so if you find, looking at the diagram, if you see that the chromosomes are seen arranged at the equator of the cell, confirm that it is metaphase. Okay, now, and it is during this phase the study of chromosomes can be done easily during the metaphase stage, because chromosomes are in the most condensed stage during metaphase. So we can easily count the number of chromosomes. The study of chromosomes is done during the metaphase stage. Then the next stage is the anaphase stage. What is actually happening during anaphase? The centromere splits. The centromere is the region where the two sister chromatids are attached. So during anaphase, the centromere splits and the sister chromatids are pulled to the opposite poles of the cell. So how does this happen? This happens by the shortening of the spindle fibers. The spindle fibers contract, pull the sister chromatids to the two opposite poles of the cell. Now next phase is the, so look at this diagram. If the sister chromatids have been separated and are being pulled to the two opposite poles of the cell, confirm that it is anaphase. Okay, next stage is the telophase stage. It is the last stage or last phase of mitosis, or the M phase, telophase. Okay, or karyokinesis. It is the last phase of karyokinesis, or it is the last phase of M phase. So during telophase, what happens? It is just the reverse of what has happened in the prophase stage. Whatever has happened in the prophase, the reverse of it happens in the telophase stage. That is, the nuclear membrane, nucleolus reappear, the spindle fibers disappear, the chromosomes or the chromosomes decondense and uncoil to form the chromatin fibers. So the daughter centrioles have, the centrioles will remain close to the reappeared nuclear membrane. So whatever has happened during the prophase, the reverse of that happens during the telophase. So telophase is the reverse of prophase. Okay, it is a stage, can be asked, the stage which is the reverse of prophase, that is telophase. Then cytokinesis. What is cytokinesis? The division of cytoplasm is called cytokinesis. You should know the difference between karyokinesis and cytokinesis. Now coming to cytokinesis, there is a difference between cytokinesis taking place in plant cell and animal cell. You should know that in animal cell, cleavage furrow occurs from the periphery of the cell, from the cell membrane towards the center of the cell. Okay, whereas in plant cell, cytokinesis occurs by the formation of cell plate. Cell plate formation takes place from the center to the periphery of the cell. That is one important thing which you should know. Okay, now you should know that centrosomes or centrioles are unique structures present only in animal cell. So if you are asked to identify whether a particular cell is plant cell or an animal cell, look at the cell, identify whether the cell, whether in the diagram the centrosome or the centriole is present. If the centriole is present, confirm that it is an animal cell. Plant cells do not have centrioles. So that is important there. Okay, so from this mitosis, you can expect a diagram based question. Any phase of mitosis can be given, and questions based on the diagram, labeling of the different parts, all these can be expected. Okay, then coming to the meiosis. The meiosis, we know it is a type of cell division taking place in the formation of gametes. So during meiosis, what happens, the chromosome number is getting reduced to half, which I have already explained at the beginning of our discussion. Then where do this, or in what type of organs does meiosis take place? In the reproductive organs. Okay, you should know that in human beings or in animals it is the testis and the ovary, and in plants you can see it occurs in the anthers and the ovules. And remember, sperms and ova and pollen grains and the female gametophyte, they are haploid in nature. You can be given a list of the four, you as the options you can it can be given: identify the diploid, or identify the cell which is not haploid, like that, and you will be given options. Okay, like for example, pollen grain, female gametophyte, sperm, skin cell. So you should know that skin cells are diploid, but female gametophyte, pollen grain, these two in plants, and in animals, the sperm cell, which is the male gamete, and the egg cell or the ovum, which is the female gamete, these are haploid. Okay, so then you should know that after fertilization, why are the gametes produced by meiosis? Because during meiosis the chromosome number is getting reduced to half. Why is this taking place? It is only if this happens after fertilization, after the fusion of the gametes, the chromosome number, the diploid number, can be reacquired. Okay, so 23 in sperm, 23 in egg. Fusion of the sperm and egg produces the zygote, which contain 46 chromosomes. So the chromosome number has been reacquired after fertilization. This can take place only if the gametes are formed by meiosis. So that is one important point to be remembered there. Now, you don't have to study in detail about the processes that are taking place in meiosis, but you should know two terms: the pairing of homologous chromosomes. What are homologous chromosomes? You should know homologous chromosomes are chromosomes of the same size and shape, one from each parent. For example, chromosome number 1 of the father and chromosome number 1 of the mother are homologous chromosomes. Another example, chromosome number 20 of father, paternal chromosome, and maternal chromosome number 20 are homologous chromosomes. So pairing of homologous chromosomes take place. When does it take place? It is during meiosis. And another term that you come across when we study meiosis is the crossing over. What is crossing over? Exchange of genetic material. Where does it take place? Exchange of genetic material takes place between the non-sister chromatids of the homologous chromosomes. So you should know what is the difference between sister chromatids and non-sister chromatids. Okay, then what is chiasma? You should know chiasma is the point or the region where crossing over takes place. The non-sister chromatids get crossed over, and the region where crossing over takes place is called as chiasma or chiasmata. Okay, that is also important. So these terms, these are certain terms which you should be familiar with from that topic, meiosis. So remember, meiosis occurs during formation of gametes. Okay, and there is a table there in our text showing the difference between mitosis and meiosis. The text which I am following is the Selena Okay, and so there is a table there, difference between mitosis and meiosis. So here you should know that table, you should study that table. You it can be asked which is not a characteristic feature of mitosis, and 3 points can be given with regard to mitosis and 1 point from meiosis can be given. For example, options which is not a feature characteristic to mitosis. Options are occurs in somatic cells, produces two daughter cells. The daughter cells produced are identical, and it is needed for gamete formation. So which is the option which is not a characteristic of mitosis? You should know that it is not involved in the formation of gametes. Okay, so you should study that table. What are the number of, or how many daughter cells are produced at the end of mitosis? You should know 2 daughter cells are produced, whereas at the end of meiosis 4 daughter cells are produced. That is also important. Okay, so these are certain things. So certain terms like chromosomes, chromatin, nucleosomes, nucleotide, centromere, centriole, centrosome, these are a little bit confusing. It might be confusing to you. So make sure that you study or you are clear with what are each of these. If you have any doubts, you can ask your doubts in the comment section. If you find this session helpful, press the like button and do comment in the comment section so that I will be back with more such videos. Stay tuned until we meet again. Thank you.